Welcome to Cell Biology, Biology 4070 for undergrads and 5150 for graduates. My name is Scott Rogers. And as we go through the class, I'd like you to, keep in mind this quote from Sir Arthur Conan Doyle as Sherlock Holmes, where he said, "The little things are infinitely the most important." And I think that that's more or less true in cell biology too, starting from very small cells with a lot of biochemistry going on inside of them, going into eukaryotic cells with multiple internal organelles, and then into multicellular organisms. You always have to think back to at the cell level in all of those. So if you need to get in touch with me, my office is 411 Life Sciences. My research lab is 515 in the Life Sciences building. This is my phone number, but often I'm not there in that particular office, so you can leave a message if you want to. A better way to contact me is through email. Just a bit about my background. My PhD is in molecular biology. My master's is in plant development. And then I have a BS in biology, general biology. My research centers around molecular biology and molecular evolution. We've also done quite a bit of work extracting microbes and nucleic acids from ice, both from glacial ice from Antarctica and Greenland, and from lake ice, including some Siberian lakes and Lake Erie. Also worked on some introns, primarily group 1 introns, nucleic acid preservation, ancient microbes and nucleic acids in pack rat middens and other dried and desiccated materials, and then also, as I said, in plant development. So I've had my fingers in quite a few little pies. So if you have any questions about any of these topics, just let me know. So there are two possible textbooks to use in here. One is Essential Cell Biology by Alberts. This is the 5th edition shown here that came out in 2019. The 4th edition and the 3rd editions are not that much different, so you could probably use either one. You can find them used, or if you want to buy a new one as well. I think they're available in hardback and paperback as well. Another book that you could use is Karp's Cell Biology, or Cell and Molecular Biology. So it's a revision of Karp's original books, and it's the 8th edition from 2016. Some classes use this one, and the two books are pretty much the same, except a few differences. But if you can get a hold of the cheapest one, either way, it would suffice for this book. I give readings in the schedule for both books. Here is an outline of the course. I will distribute videos, YouTube videos, and then during the class time we'll discuss those videos, that information. So that'll be on Mondays and Wednesdays from 10:30 to 11:20 in room 334, Life Sciences. And you must view the videos before you come to class, otherwise you won't have much to discuss. Also on Thursdays from 9:30 to 1:20 in room 445, Life Sciences, we'll have lab, and I'll have a short lecture to introduce the lab at 9:30 in each case, and then you'll go and work in groups, about four students per group, and so you'll have to cooperate with the people in your group, and everyone should participate equally in those groups. There will be 3 exams or quizzes. The first one will be handed out on September 30th, and then October 28th and December 9th. And these are all take-home exams, so you'll have about a week to complete them. For each exam, you will be given 6 questions. and then you will choose 2 of those to answer in detail. They're all essay, drawing explanation, no multiple choice questions in this course. And I'll give you about 4 weeks before each exam, I'll give you a set of practice questions, and some of these will actually be used in the exam itself. The exam is designed to be a learning exercise, not a testing exercise. Also, there will be a minimum of 2 pages single-spaced for each of your answers, the answer to each question. So you need to, for each exam, if you're answering 2 questions, that you'll need to write at least 4 pages for your answers. 2 pages for each of your answers. Things like cheating and plagiarism, etc., will equal a 0, and every year I get a few people trying to do that. It's easy to pick up, and that will mean that for that particular exam, that you will get a 0 for that exam, and so that's 1/3 of your grade already, which is putting you down in the C to D range right away. Also, for lab, you'll be required to keep a lab notebook, and they need to be updated immediately every day you are in lab. You don't wait to write in those at a later time, because you'll forget many things and they'll never be written down. So in your lab notebook for each lab exercise, you'll have to write the date, the lab exercise, any notes, diagrams, and conclusions for that particular lab. Also, since you'll be working in groups and sometimes the labs don't fit neatly into the time allotted on Thursdays, you might have to get some information going back and forth with your lab mates as well. Also, because we're still under the threat and rules governing activities during COVID-19, you will be required to wear face coverings while in the lab and in the classroom, as well as anywhere in the building. There are more details about this in the syllabus, so be sure to read those details. If you would like more detail about what I'll be covering in the class, I wrote a book that was published in 2017 called Integrated Molecular Evolution. This is a book I use in my class called Molecular Evolution, but if you want more information now while you're taking cell biology, this is a good resource. Also, another book I wrote that was published in 2020 is called Defrosting Ancient Microbes, and that discusses the isolation of microbes from glacial ice, as well as nucleic acids from glacial ice, including some pathogens. It goes through quite a few details, but it's actually a trade book, so it's meant to be read by a general audience, so it should be rather easy for you guys to read this. So now I'd like to give you a little pre-test, and you won't be graded on this or anything, but just to try to determine what you know, what you think you know, what you don't know, things like that. So I'll discuss the answers to these in class, but right now, if you just look through these, you can probably start and stop the video. But question number 1 is, how many cells are there in the average human? How many total cells do you have in your body? And what are they doing, by the way? Second is, how many bacterial cells are in and on a typical human? And a lot of them are in your gut, which help you with digestion. What are the dimensions of an average bacterium? And then what are the dimensions of an average eukaryotic cell? So here's page 2. How many ribosomes does an average eukaryotic cell have? That is, in your cells, how many ribosomes, which take the mRNA and translate the message into a protein or polypeptide, how many of those does an average eukaryotic cell have? So question 6, chloroplasts produce ATP and NADPH from sunlight by splitting what? So ATP is used for transferring mainly energy, bond energy, and NADPH is primarily for shifting around electrons. But what is being split during photosynthesis? 7. Mitochondria produce ATP and NADH. By what process? So they're also making ATP, and in this case NADH. And what process is being used to produce those? Okay, and then most cancer cells have defects in their what? What kind of pathways or enzymes do most cancer cells have defects in? Question 9. Where does the carbon dioxide that you exhale come from? Right? So you breathe in oxygen, it goes through this part of this process of oxidative phosphorylation, and then you exhale carbon dioxide. So where does carbon dioxide come from in that entire process? And by the way, there are some organisms that can take the carbon dioxide and reverse that process and fix the carbon dioxide into carbon-containing organic compounds. Question 10. In anaphase of mitosis, what are being pulled apart and are moving towards each pole? And for comparison, question 11. In anaphase 1 of meiosis, what are being pulled apart and moving towards each pole? And then finally, in anaphase 2 of meiosis, what are the things being pulled apart and moving towards each pole? Okay, so that's a little pretest. Have your answers ready for class. Okay, now we have to talk about what cells do and what they need to do to survive and communicate with other cells. So the first question is, why do you eat and drink? Well, the simple answer is it tastes good and it's good to drink something. But the real reason is that your brain is telling you to eat and drink certain things. And the same thing with bacteria. They have to gain nutrients from somewhere, gain moisture, water, etc. from somewhere. So that starts very early in the evolutionary process. And also, why do sweet things and salty things taste good to you, but bitter things taste bad? And why do we have taste at all, right? Well, one of the reasons is sweet things usually contain glucose. Your cells need glucose. Salty things contain sodium and usually potassium and other ions that you need. So you're eating those because your body requires them, and you have to taste those to make sure that you're getting the proper things into your body, and that's been an evolutionary process also. Bitter things and things that are very acidic usually don't taste good to us, and that's because many of those are toxic to us. And so the really bitter things that you taste occasionally, it's really a signal that's been evolutionarily programmed to tell you that, oh, these are bitter things, they could be toxic. So here are a few of those M&Ms. You can sort of taste those right now as you're looking at that. And hamburger, you can taste that salty thing that you put in your mouth. And then you look at grapefruit and you go, that's a little bitter. And that's why if you give something bitter to children, they give this kind of face. And so that's been, the process of evolution, indicating that those things are probably not good for you. At least most of those are not good for you. So usually the organisms that could not sense these things and ate them anyway, many of them died, went extinct, and the ones who had a certain taste or a certain sensation when they ate something or came in close contact with something, and it was good for them, then they survived and went on to reproduce and form more of those types of cells. So in general, what do cells do? And by that I mean almost all cells have to do certain things, otherwise they don't survive. So all cells have to either synthesize or sequester and use compounds with carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, as well as some other elements. They also need to use glycolysis to make some ATP in the fermentation process. The end product of glycolysis, in many cases, is pyruvate, and that is then shuttled into the TCA cycle, the tricarboxylic acid cycle, which takes that pyruvate, joins it with other carbon-containing compounds to produce more NADH and more ATP. The NADH is then used in oxidative phosphorylation, which essentially uses oxygen as the electron acceptor plus hydrogen to make water. But in doing so, that creates a lot of energy. The energy from that is used to pump protons across the inner mitochondrial membrane, which then those protons flow back through ATP synthase to make a great deal of ATP. So other cells, including bacteria, plants, and some protists, are able to use photosynthesis to create their carbon compounds. So they can fix carbon dioxide and make carbon compounds using that carbon to then store things like glucose, starch, and other compounds. Many organisms can do the reverse of glycolysis, that is, take pyruvate and some other carbon compounds and reverse the process to make glucose, and then the glucose can be used for other metabolic functions. And then they have other metabolic and catabolic functions, including fixing carbon in photosynthesis. But that's not the only way to fix carbon. You can reverse the TCA cycle in some organisms, and then there are other ways of carbon fixation as well, and also nitrogen fixation, another set of genes and proteins that allow certain organisms to fix nitrogen into more complex molecules. So if you look at the periodic table of elements, organisms use a limited number of those elements. So the primary ones are in red here: hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. And you find all but sulfur in nucleic acids, and then you find in proteins, you find sulfur in addition to that. There are certain ions that are used, including sodium, potassium, magnesium, calcium, and chlorine, the primary elements that are used as ions. There are other ions too, which are molecules. And then there are these ones in blue here, which are secondary elements. Iron is one that's used quite often. Copper is used in a lot of different, as a cofactor in many different proteins, as in zinc and silicone and aluminum, etc. And then there are some that are toxic because they are close in their activity to some of the other elements, and so those become somewhat toxic. However, selenium, this one here, which is listed as toxic, actually is used by some organisms, including humans, as a substitute for sulfur in some proteins. So at very low levels, it's actually beneficial. If you don't get any selenium, it can be deadly, and if you get too much selenium, it's also toxic. And if you break those molecules down a little more, they're really made up of protons and neutrons and electrons. So a proton is simply a hydrogen atom without its electron. So that's a proton. Very simple element. And then you have protium, which is a hydrogen atom, and then deuterium, which has a proton and a neutron, so it's twice as heavy, essentially heavy hydrogen. And tritium, which is radioactive, has 2 neutrons and a proton as well as an electron. And then you go to the helium isotopes, which are the common ones are helium-3 and helium-4 here. So you're essentially adding a number of protons, a number of neutrons, and then electrons as well, in addition to the ones that you normally find in hydrogen. So you can sort of build these things up atom by atom by adding protons, neutrons, and electrons. So very early on in the formation of the universe, first there were quarks, and then finally there were hydrogen atoms and helium atoms. But how did the other elements form, especially the ones that are biologically relevant? So in the early stars, the sun and other stars, then you push these protons together. You get first hydrogen and deuterium. Those can bang together and you can get helium-3, and then you have energy given off in the form of gamma rays and other and light energy, etc. Then the helium-3s can bang together and form helium-4 and some hydrogen. Heavier atoms can be formed also by joining two helium atoms together to form beryllium and lithium, and then there are even more processes that are produced by exploding stars, as well as the high pressures in the centers of the stars, to make things like carbon, nitrogen, oxygen, and some other elements. So here's another picture of the periodic table of elements showing the origin of each one of those elements. So the primary ones that would form in the Big Bang fusion and in stars like the sun would be hydrogen and helium, so you get those happening right away. Then you have other things occurring, dying low mass stars, cosmic ray fission, merging neutron stars. Took more energy to get these elements that are heavier and heavier, pushing more protons and more neutrons together. And these ones in here, exploding massive stars. So when Carl Sagan said we are star stuff, it's really true that most of the elements that we need, these ones over here, carbon, nitrogen, oxygen, etc., were from dying stars, low mass stars, as well as exploding massive stars, and then those original hydrogen and heliums fusing together to make these larger and larger and heavier atoms. So as far as the sun also, because as you go further and further into the sun, there's more and more pressure. So all of the elements from hydrogen all the way up to about iron can be produced, nickel and iron can be produced in things like the sun and other stars, because you're getting pressure, more and more pressure as you move towards the center of the sun, and these elements are getting heavier, but it kind of stops there. And so the only way that you can get the elements of higher molecular weight, higher mass, is by exploding stars and neutron stars and things like that. Much more energy is required to make those larger atoms. So here's a little continuum of what happened to form the universe and eventually the Earth. So about 3.75 billion years ago, there was the Big Bang. First there were quarks, and then hydrogen and helium. Then stars started to form, and finally galaxies started to form. Here's our Milky Way galaxy. And on one of the minor arms of the Milky Way galaxy, this puff of stuff started accreting into what we now have as the solar system. And so the solar system formed about 4.5 to 4.6 billion years ago. And what I mean by the stuff in that ball, there were things like iron and carbon and oxygen and hydrogen, all the things necessary for life to start on Earth. And there may have even been amino acids and hydrocarbons and other things that also led to life on Earth. And then just as the Earth was beginning to solidify, it was molten before, then a huge Mars-sized body hit the Earth. Possibly it was becoming another planet, but it hit the Earth and kind of a glancing blow. And in doing so, it melted the entire surface of the Earth, but also it spewed off a great deal of the Earth and part of that body that hit the Earth and congealed into a moon, or actually initially more than one moon. And they were actually very close to the Earth at that time, so it would have almost filled the sky at that time, 4.5 billion years ago. And so that created great tides on the Earth, not only of water, which wasn't quite there until about 4.3 billion years ago, when there were oceans forming already, but it also distorted the Earth's crust, and so it created a lot of tectonic movements and volcanism. So at that time, the Earth was very much hotter. There was a lot of volcanism, a lot of water vapor and sulfur in the air. It was a reducing atmosphere, almost no oxygen, and so it was very unhospitable to most organisms, so that life was probably not present at all 4.5 to about 4.2 billion years ago. So then it's clear that all of this created quite a bit of havoc on the Earth, and the Earth almost was totally obliterated by this collision, but enough of the surface of the Earth remained intact, and it actually gained in mass, because most of the body that crashed into the Earth was retained on the Earth, but a great deal of the mantle material from the Earth actually became incorporated into what is now the moon. And as I said originally, there were 2 moons, and there's actually more debris still floating around out there that hasn't incorporated into either of those moons. But then very shortly after that initial formation of those 2 moons, one caught up with the other, and in sort of slow motion it kind of melted into the moons. And the reason that was first theorized is one half of the moon has all of the highlands, and then the other half of the moon has all the lowlands, and so it was thought that a major collision between 2 bodies occurred, and so you have this high area on one side, which is formed from the one smaller moon essentially melting into the other larger chunk that became the moon that we know today. And as I said, the moon was much closer in those days, so it created more tides, eventually with the oceans, but also with the Earth's crust. And it's still moving away from the Earth, so the moon is currently moving away. Also, a day length on the Earth was about 6 hours long, so the Earth itself has been slowing down in its rotation over the past 4.5 billion years. So in essence, for the past 4.55 billion years on the Earth, nothing has really remained the same. It's constantly changing. So that creates a lot of opportunity for various organisms, but it also presents some major challenges for organisms as well. So here are 2 artists' renditions of some times on Earth. This is in the Hadean, 4.55 to 3.8 billion years ago. So early in that time, there was mainly a molten surface, although by about 4.2 to 4.3 billion years ago, there were oceans already. So there were many areas on the Earth where the crust had solidified. So there was an opportunity by about 4.2 billion years ago to 4.1 billion years ago to life, that is cellular life, to begin growing on Earth. By the Archean eon, 3.8 to 2.6 billion years ago, a lot of life was present. So although there was a reducing atmosphere, so these were all anaerobic organisms. And all bacteria at that time, bacteria and archaea. So the bacteria first can be recognized as being on the Earth by about 3.8 to 4 billion years ago. And the archaea, some new reports say that they were around as early as 3.4 billion years ago. And then by 2.6 billion years ago, there is one branch of the archaea that will eventually lead to the eukaryotes. But it wasn't only what was on the Earth at that time that led to life. Things were arriving from space in the form of comets and asteroids, bringing things like hydrocarbons and amino acids and purines and pyrimidines. So some of the basic units of life were brought to the Earth extraterrestrially very, very early in Earth's history. So if you take amino acids and add heat and pressure, such as the heat and pressure of a comet entering the Earth's atmosphere, then you can get small peptides, so you can get more complex things forming. And those, as well as hydrocarbons that are, sort of look more like fatty acids, you can get those forming as well. So these things being brought from the outside of the Earth, as well as things on Earth already, led to a lot of different compounds that were important to the development of living systems on Earth. Volcanism also led to a lot of things a lot of compounds, including water vapor, that would spew out of the volcanoes. Not only the smoke and the ash, but there's a lot of steam that comes out of volcanoes, and so a lot of the cloud formations and things like that came from volcanoes as well as from comets too. So a lot of the water on Earth was from those two major sources. Also in the oceans, in these areas where you have thermal vents, there are many different chemicals released by the thermal vents, and the heat is also there, and the pressure. And so this can lead to the formation of many different biologically relevant compounds, including amino acids, but also carbon compounds as well. So there are many theories that life originally arose on Earth in these deep ocean vents rather than on the surface. On the surface, the problem is that there's a lot of UV radiation, which will essentially destroy any biological molecules that are formed. So it's probably likely that most of the life formed very early in deep ocean vents. Here are some pictures of a couple of deep ocean vents. These are called black smokers because they have a lot of black material coming out of them, usually sulfur compounds. You can also see life around here. So these are a specific type of clam that are known to live around these black smokers, and so they're getting nutrients from those as well as energy from the heat of the volcanic activity. So there are a couple of different really important parts to this. One is that the heat of the water and chemicals coming out of here is about 400 degrees centigrade, whereas some distance away the temperature of the water is about 4 degrees centigrade. So there's a big temperature gradient across there, which would allow for different types of organisms to survive at specific temperatures where their physiology permits. Also, from the inside tube of those black smokers to the outside, there's a pH gradient, and so you have several different things occurring here. There's nutrient gradients, pH gradients, and temperature gradients across here, which would favor the origination of different species and survival of different species in different locales around there. So a lot of different microenvironments around those black smokers and other types of thermal vents. Then when you start looking around the Earth, you find that there are many types of environments and habitats available to a wide variety of organisms. So the land area is 148 million square kilometers now. The crust is 5 to 10 kilometers thick, and there have actually been found living communities of bacteria that are about 5 miles, so about 6 kilometers or so into the Earth's crust. Then the ocean is about 361 million square kilometers, but the volume is 1.3 billion cubic kilometers of water. The atmosphere's depth today is about 100 kilometers thick, although it gets very, very thin at those high elevations, and a total volume of 4.2 billion cubic kilometers of the atmosphere. Although if you look from a distance, our atmosphere is very, very thin, a thin little layer over the surface of the Earth. But in short, there are many, many different places where organisms can live based on the proteins they make and their physiology, whether they need the sun or whether they need air, oxygen, or to be out of oxygen. So a lot of different places for organisms to live and survive and evolve on Earth. So there are several things that have caused some major changes in evolution on Earth. One is frequent impacts with asteroids and meteors. So those, especially the large ones, have caused huge changes in the climate on Earth, and the conditions on Earth, and the extinction of organisms. Oxygenation of the atmosphere, which started about 2.7 billion years ago, before there was a reducing atmosphere, before 2.7 billion years ago. But then oxygenic cyanobacteria got active, started growing in the oceans, and then increased gradually the concentration of oxygen in the atmosphere. So today we have about 21% of the atmosphere is made up of oxygen. It's been higher sometimes it was up to 35% oxygen in the atmosphere. And then there were these endosymbiotic events, one leading to the formation of mitochondria from a proteobacterium that could do oxidative phosphorylation. This helped the protist cell now with a nucleus, which probably came from a different type of bacterium, a planktomycete. This then gave that cell the capability to get a lot more ATP out of each glucose molecule by using oxygen to join with hydrogen and electrons to generate a lot of energy, and then to glean off that by using certain enzymes and proton pumps to make a lot of ATP. Another innovation was to, in plant cells, to take in a cyanobacterium that became the chloroplast to do photosynthesis, thus changing a protist cell from a heterotroph into a photoautotroph, meaning that it could now take carbon dioxide from the air and start fixing that to other carbon compounds to make all of the organic structures needed for those cells. Then the last one over here is multicellularity. So possibly about 2 billion years ago, some of the first cells started to cooperate with other cells of the same species, of the same type, and become a multicellular organism. And then that greatly expanded about 1 billion years ago to become much larger, more complex multicellular organisms. So here's a brief timeline of Earth history. Formation of the Earth was about 4.55 billion years ago, and then shortly thereafter the formation of a moon caused by collision with a Mars-sized body, first to form a couple of moons, and then those fused into one larger moon. And the moon's been moving away since then. Partly solid Earth here, and probably even partly solid even before that. Some of the oldest rocks that can be found are about 4 billion years old. The first life in terms of a membrane-bound organism that probably was mainly based on ribozymes and ribosomes. It's put here at 3.8. More recent evidence says probably 4.1 billion years ago, or maybe even older than that, for the first RNA-based organisms. Then the evidence of the first bacteria, this one shows 3.5 That's been moved back a bit too, but to about 3.8 billion years ago, and the first archaea were about 3.4 billion years ago. Then the oxygen in the atmosphere started to increase due to oxygenic photosynthesis of cyanobacteria. The first eukaryotes, probably about 2.4 billion years ago, and mitochondrial eukaryotes, 2.2 to 2.4 billion years ago. And then the beginning of the plants occurred about 1.9 billion years ago. Then you get fungi, which this number's been moved back to probably to at least 1.5 billion years ago. And on this chart it says first multicellular eukaryotes about 1 billion years ago, but there were multicellular bacteria back probably as far as 2 billion years ago. And then you have this area here where it seems like things are more concentrated, and that's primarily because we can find more of those as fossils, including land plants, animals, ferns, sharks, gymnosperms, etc., dinosaurs, angiosperms, and finally Homo sapiens at way at the end. So then let's start talking about what a cell is. What is a cell here? So we can always say, well, it's surrounded by a membrane, a double membrane on the outside. This includes bacteria, archaea, and eukaryotes, and then it has some sort of nucleic acid inside, and ribosomes, and things to make different proteins and to alter those proteins in certain ways. So we can sort of define a cell in many different ways. But before I get to that, I want to discuss the sizes of these things inside of a cell. So if you take a single cell, in this case a plant cell, and you blow it up to room size, then start placing things in there, here's what you'd find. So here's 2 people in that room just for reference. So the plant cell wall would be on average about the thickness of a wall in a house or a building. And then surrounding that inside you'd have a membrane, a phospholipid membrane, that would coat every surface of that room. And embedded in the membrane, some going through the membrane, and also some just sticking to the surface of the membrane, you'd have a number of different proteins. In the middle of the cell somewhere, you would have a nucleus, which is surrounded by 2 membranes, but it's actually a single membrane folded over on itself. And then nuclear pores coating that, which would allow some proteins to get in and RNAs to get out of those nuclear pores. And of course, inside the nucleus, you would have the DNA. And for example, your DNA, one chromosome, would stretch for about 20 kilometers, an average chromosome. And you have 46 chromosomes, or 23 pairs of chromosomes. So if you multiply that out, you have a long set of genes, chromosomes, within your nucleus. And many plants have about the same amount of DNA in their nucleus, and some have a lot more than that in their nucleus. So imagine having to fold up all of that DNA, which on our scale would be about the thickness of twine, trying to fold all that up into something that would be about 2 meters in diameter, this nucleus. And also replicate that and keep it in some organized fashion for expression, and replicate and separate it. So a big feat to do with that much DNA in the nucleus. Also within the nucleus is an area called the nucleolus, which is not membrane bound, and that's where the ribosomal RNA is transcribed, and the ribosomes are assembled into the large and small subunits before they're shipped out of the nucleus. Transcription also occurs in the nucleus, so you'd have these RNA polymerase molecules moving down the DNA, producing an RNA copy of that region of the DNA, and it would be about the thickness of string. And then that would be transported out of the nucleus and onto either cytoplasmic ribosomes or ribosomes on the endoplasmic reticulum. And the ribosomes would be about the size of a golf ball. And then a large protein would be about the size of a lima bean. A small protein being produced there would be about the size of a pea. And an atom, you could actually see an atom as just a sort of a dust speck in these cells. Mitochondrion in here would be about the size of a person, sort of a fat person, and that would be doing the oxidative phosphorylation. Chloroplast would be quite a bit larger than a person, probably 3 or 4 meters in length and a meter or so in diameter or in thickness. If there were bacteria in the cell, they would be also about the size of a mitochondrion, although there are some small bacteria that are more on the size of these viruses down here. So viruses would be much smaller, although again there are some viruses that are as large as some small bacteria. So imagine now there are multiple mitochondria, multiple chloroplasts, multiple viruses, only one nucleus, but also about 2 million ribosomes and millions of other proteins squeezed into that room. In fact, cells are so packed with proteins and other molecules and ions and things like that they are nearly crystalline, so just really packed into that cell. And now imagine that big room being shrunk down to its actual size, and this would be to squeeze it by 1 million times until it was barely visible on your desktop, and then you need to squeeze it even further than that. In fact, you can see as a little speck about 25 cells. So imagine 25 cells being packed together in that way, and you could actually see those on your desk or on your sleeve or wherever you want to find them. So that's the actual size of those cells with all of those components packed into each one of those cells. Also, be aware that all cells are not the same size. There's quite a bit of variation even among bacteria. So here's Rickettsia bacterium down here, and here's an E. coli. There's a red blood cell. You can just see the edge of it there. Here are some others. There's a Mycoplasma bacterium, Haemophilus, Staphylococcus, E. coli, Bacillus, and then here's a red blood cell again. And you can see also viruses. Some viruses are as large as some small bacteria. And then you get into smaller and smaller viruses, including poliomyelitis virus down here, a very, very tiny little virus. So here's a typical gram-negative bacterium with 2 membranes and a cell wall in between them, made of peptidoglycan. There's a nucleoid, which is the DNA with the protein, DNA binding proteins bound to it. A lot of ribosomes. Usually in a cell like this, you'd have 20,000 to perhaps 60 or 80,000 ribosomes in a cell. And there's a flagellum, some pili, and simpler than a eukaryote, but already very complex biochemically. So here's a typical animal cell where you have a cell membrane, there's a cytoplasm, there are some mitochondria, a nucleus with the rough and smooth endoplasmic reticulum, the rough ones containing ribosomes, and then there are cytoplasmic ribosomes. Remember, there's about 2 million on average, 2 million ribosomes per cell. And it's not shown clearly here, but there are proteins embedded in the membrane, and those are then able to communicate, the communication between one cell and the next in these eukaryotic cells. And here's a typical plant cell with the cell wall and then the cell membrane inside of that. Now, in addition to mitochondria, you have chloroplasts and a vacuole, which is a large organelle which controls movement and concentrations of different things in the cell, and also maintains turgor pressure in this cell, which can't move a lot because of the cell wall. You also have these plasmodesmata that connect one cell to the next, and those actually connect the cytosol of one cell to the other. So there's a continuous cytosol within plants and also in fungi, which do it that way, which is different than in animals, which have just cell surface proteins that communicate back and forth with the adjoining cells. So just a few facts to get you started thinking about numbers. When did the first cells appear on Earth? Probably 4.2 billion years ago, although they were based on RNA and not DNA. So RNA plus peptides rather than DNA polypeptides or proteins and other things. And so the true bacterial cells based on bacteria probably appeared around 3.8 billion years ago. How many cells in the human body? About 37 trillion. A lot of cells. And those cells have to communicate with other cells. And so if they don't, then you have a problem. So how many cell types do you have in your body? Over 200 different types of cells. How many neurons in the humans? And that's about 8.6 times 10 to the 10th. That is 86 billion neurons. That's 2% of the total number of cells in your body. So that's another way to communicate cell to cell, and that's those are the neurons. How many in the cerebral cortex? About 1.9 to 2.3 times 10 to the 10th. And that's about 22 to 27% of all the neurons are located in the cortex. So how many bacteria do you have on and in you? And that's about 4 times 10 to the 13th. So that's actually more bacteria than you have human cells. And if you look at the number of viruses, there's probably 10 times that number. How many cells are in the nematode Caenorhabditis elegans? Exactly 959, including 302 neurons, so quite a few neurons there. And eggs and sperm are additional because they're constantly being formed. How many cells in a giant sequoia tree? About 2 million trillion, which is equal to 2 quintillion. So many cells in those giant trees. In fact, as you can probably tell right now, it's unimaginable. You can't really imagine that number of cells. All of life on Earth is joined together in one continuous string of changes in DNA and then eventually the proteins and the physiology, etc. So about 3.8 to 4.2 billion years ago, you have this root which is made of RNA molecules, at that time RNA organisms. And then about 3.8 billion years ago, you get these precursors to what become the bacteria, which originated 3.5 to 3.8 billion years ago, and then started diverging first into the monoderms, the gram-positive bacteria, and then eventually into the gram-negative bacteria, which are the diderms. They have 2 membranes. And then about 3 to 3.4 billion years ago, you get the first archaea, and those started diverging into the Euryarchaeota and others. And then one branch of this branched off and joined with a bacterium over here, probably 2 different bacteria, to become the eukaryotes. So eukaryotes can sort of be defined as these cooperating archaea and bacteria, cooperating as endosymbionts. So your cells and all animal cells are partly archaeal in origin, and partly a planktomycete in origin, and partly a proteobacterium in origin. So those are all 3 different lineages of organisms coming together to make up this group called eukaryotes. And then plants have picked up another bacterium, a cyanobacterium, and then these protists have picked up some of the plant cells inside of them to become photosynthetic. So eukaryotes are very good at forming these endosymbiotic relationships to rapidly improve their condition or their survivability. This figure is a different phylogram of the same types of organisms, but it illustrates the degree of unicellularity in a group versus multicellularity in a group. So you can see most bacteria are unicellular. There are a few that are multicellular. Same with archaea, and then these basal groups of protists, including the excavata, acrobians, rhizaria, alveolata, and stromatophiles. Then you get to this sort of what's called a crown group, where the multicellular versions predominate. So in the plants here, Archaeplastida, the amoebas here, fungi, and animals, most of the members are multicellular, whereas some of the basal groups way down here near the base of each one of those parts of the tree, each one of those leaves, those are the unicellular, sort of the pioneers of each one of those groups. So if we talk about archaea and bacteria, often people refer to these as prokaryotes, meaning before or without a nucleus. So it's the definition from the internet is a microscopic single-celled organism that has neither a distinct nucleus with a membrane nor other specialized organelles. So prokaryotes include bacteria and archaea. But this turns out not to be a great definition and not universally true. So if someone said, show me a bunch of bacteria, they would probably do this. Here's E. coli, Equifex, Aeolicus, Colobacter, Spirochetes, Microcystis, and Mycobacterium. And all of these don't have internal membranes, and they don't have a nucleus. So that fits generally with that definition of prokaryote. But that doesn't quite fit the definition of this member of the planktomycetes. So this is Gemeta. And it has a double membrane surrounding what is the nucleoid, the DNA. So it looks sort of like a nucleus. It also has little sections along that membrane-bound structure that look like nuclear pores, and ribosomes stick to that as they do in eukaryotes as well. So this looks like internal membranes and specialized organelle structures, and so that doesn't fit with that definition of prokaryote. There are other planktomycetes that have even more membrane structures, not necessarily this nucleus-like structure, but other membrane-bound organelles. And so this one group, this may be where the nucleus and the nucleus came from in eukaryotes, as well as internal membranes, because the planktomycetes do have a nucleus-like structure, and they have internal membrane-bound structures that look much like organelles. And so this is one group that kind of defies that prokaryote definition. So I think the term prokaryote is probably outmoded and not very accurate. Literally, prokaryote means before a nucleus or without a nucleus. And it's sort of inappropriate because normally you don't define things by what they do not have. So you define them by what they do have. So you can define a human as an organism without fins. Yeah, that's true, but that's not really all humans. And trees don't have fins, so are they human? So you can't really use a definition that is a negative in that way. Also, as I just stated, some planktomycetes appear to have an organelle that resembles a nucleus, as well as other truly membrane-bound organelle structures. So in this course, I'm going to use prokaryote sparingly, and I hope you will too. Instead, I'll refer directly to bacteria and archaea, and also, of course, to eukaryotes. So here are a few archaea, and on the surface they sort of resemble bacteria, and originally they were just a different group of bacteria. But now we know they're quite removed from bacteria, have a lot of different molecular structures that are closer to eukaryotes than they are to bacteria. And so they're definitely a clearly defined group separate from bacteria. So again, here's another definition from the internet. And eukaryotes are organisms consisting of a cell or cells in which the genetic material is DNA in the form of chromosomes contained within a distinct nucleus. And again, planktomycetes also have something like this. Eukaryotes include all cellular organisms other than bacteria and archaea. I don't necessarily like that statement, but anyway, the way I think of eukaryotes is they originated from the joining of one archaeal cell and at least two bacterial cells, and sometimes more than two bacterial cells, and then spread out from there. And in fact, some of the eukaryotes have sort of gobbled up other eukaryotes, and those have become endosymbionts in those, and so there are even more complex members of the eukarya than the simple ones such as us. And here's one strange eukaryote. This is Giardia lamblia, and if you happen to ingest this, you'll get diarrhea, so it is a pathogen. It has no mitochondria, but it has two nuclei, so it sort of looks like something that wouldn't quite fit most definitions of a eukaryote. But it's truly a eukaryote, and an amitochondrial eukaryote. That is, it has no mitochondria. And that harkens back to a time when the first eukaryotes originated. It was probably the combination between a planktomycete and an archaeal cell. Neither one of those has oxidative phosphorylation, and so later a cell somewhere along the line picked up a proteobacterium that did have oxidative phosphorylation, and that led to the formation of the mitochondria. So we'll talk about many of these characteristics of cells during the course, as well as some exceptions to some of the rules, and there are a lot of those. Okay, that's it for this time, and I will talk to you later.